Antarctica's iceberg A23A had been stuck to the ocean floor for decades, but it broke free last November, gradually heading north towards open water. The size is enormous, with a surface area of about 1,500 square miles.
12 days. That's all it took. 12 days after a chunk of ice the size of Chicago snapped off the edge of Antarctica, scientists sent a robot down into the freezing darkness beneath it. The cameras switched on, and what appeared on those monitors silenced an entire research team. Not because the seafloor was empty, because it wasn't. Life was everywhere, and it had been living in the dark for centuries, completely cut off from the sun, from the surface, from everything we thought life needed to survive. The Iceberg event. To understand what scientists found, you first have to understand the scale of what broke free. On January 13, 2025, an iceberg designated A84 calved from the George VI ice shelf along Antarctica's western coast. A massive floating glacier attached to the Antarctic Peninsula ice sheet in the Bellingshausen Sea. This wasn't a crack or a sliver. This was a catastrophic fracture. Iceberg A84 measured approximately 510 square kilometers, roughly 19 miles long, 11 miles wide, and extending some 500 feet downward into the ocean, roughly the size of Chicago. NASA's Terra and Aqua satellites captured the moment in satellite imagery, a vast slab of ancient ice peeling away from the continent and beginning its slow drift through coastal currents. Scientists watching from orbit could see it clearly. 84 was moving, bouncing against stretches of the Antarctic coastline as ocean currents carried it away, carving its own path through some of the most remote and hostile waters on Earth. But here's what made this different from every other iceberg that has ever broken free from Antarctica. It wasn't what A84 was, it was what A84 had been covering. For potentially hundreds of years, possibly longer, that slab of ice had sat on top of a section of the Antarctic seafloor, pressing down with the weight of a skyscraper made of frozen water. Beneath it, absolute darkness. No sunlight penetrating 150 meters of solid ice. No photosynthesis possible anywhere in that sealed column of water. Surface nutrients completely cut off. The water temperature down there hovered near freezing. The pressure at depth would crush the hull of a submarine. Every textbook in marine biology would have told you the same thing. That seafloor should be nearly lifeless. Sparse organisms at best, microbial life clinging to existence in the sediment. Maybe a few hardy creatures scraped into survival by geological accident. That's what the science predicted. That's what every model said you'd find. But the moment A84 broke free, the clock started. Because somewhere in that vast, frozen southern ocean, a research vessel was already in the water. And they were about to make a decision that would change everything science thought it understood about where life can exist on this planet. The lucky pivot. You have to appreciate just how thin the margin was here, how close humanity came to simply never knowing what was underneath that ice. The Schmidt Ocean Institute's research vessel, RV Falkor 2, was already operating in the Bellingshausen Sea when A84 calved. The team aboard was conducting deep-sea biodiversity research, collecting sediment samples, analyzing ocean currents, mapping the understudied floor of one of Antarctica's most remote coastal regions. It was important work, meticulous work, the kind of scientific expedition that rarely makes headlines but slowly builds the foundation of what we understand about the deep ocean. Then, they hit a problem. Unexpected sea ice was blocking their originally planned route, forcing the vessel to restrict its operations to a smaller area than the team had intended. Frustrating, but not unusual in Antarctic waters. Ice here doesn't ask for your permission. You work around it. Then word came through about A84. Dr. Patricia Esquete from the Center for Environmental and Marine Studies at the University of Aveiro remembered the moment clearly. The decision, she said, was immediate. The entire expedition plan was thrown out. The team pivoted, changed course, and went for it. They had a window, possibly the only window that would ever exist, to reach a stretch of seafloor that had been sealed from the surface world for potentially centuries. Every hour they delayed was an hour the ecosystem beneath would begin adjusting to its new reality. 12 days after A84 broke free, RV Falkor 2 reached the newly exposed seafloor. In the history of Antarctic exploration, this was an extraordinary alignment of circumstance. A research vessel, already in the right ocean, already equipped with the right technology, already carrying scientists with the exact expertise needed, pivoting on 12 days' notice to reach one of the most remote and newly accessible locations on Earth. Dr. Giotica Virmani, executive director of the Schmidt Ocean Institute, put it plainly. Being right there when this iceberg calved presented a rare scientific opportunity. Serendipitous moments, she said, are part of the excitement of research at sea. They offer the chance to be the first to witness the untouched beauty of our world. Good luck played a huge role. The team acknowledged that openly. But there's a lesson in it too. You can only seize the lucky moment if you're already out there, already in the water, already doing the work. This discovery didn't happen in a laboratory. It happened because people chose to be in one of the most inhospitable places on Earth. They were about to find out exactly why that choice mattered. Descent into the dark. The instrument that would make history is called ROV Sebastian, a remotely operated vehicle built and operated by the Schmidt Ocean Institute. It's tethered to the research vessel by a cable that snakes down through hundreds of meters of water, transmitting live high-definition video back to the monitors on the ship above, while pilots guide its movements through the crushing dark. It carries cameras, sampling arms, sensors capable of measuring temperature, salinity, pressure, and biological density. It is, in every practical sense, the eyes of science in a place no human body could survive. On January 25, 2025, 12 days after the calving, 12 days after the team had pivoted their entire expedition, ROV Sebastian was lowered over the side of RV Falcor 2 and began its descent, down through 500 feet of water, where the ice shelf had sat for centuries, down through the cold, pressure-thickened darkness, down to depths reaching 2,300 meters in some passes, with early exploration beginning around 1,150 meters. The cable played out. The ROV's lights illuminated nothing but particles drifting in the current. Marine snow, flecks of biological material suspended in the water column. Standard, expected, unremarkable. Then the seafloor came into range. The cameras focused. The images sharpened on the monitors in the ship's control room. Scientists leaned forward. Somebody gasped. The seafloor wasn't barren. It was covered. Not sparse, not barely populated, covered. Large organisms clustered across every surface the cameras could reach. Structure everywhere. Vertical growth rising from horizontal rock, movement along the sediment, creatures drifting through the illuminated water column in every direction the ROV turned. For 8 consecutive days, ROV Sebastian explored this newly exposed landscape, covering sweeping transects of the seafloor, filming continuously, collecting samples with its mechanical arms. The team watching from above barely spoke. Dr. Esquete later described it as one of the most surreal experiences of her scientific career. Not just the discovery, but the silence that fell over a room full of trained experts who simply didn't have words for what they were looking at. Because what was on those monitors didn't match the prediction. It didn't match the textbook. It didn't match anything. Life had not merely survived here, life had thrived. And now the cameras were about to reveal exactly what form it had taken. Creature by extraordinary creature. The creatures. Start with the sponges, because the sponges alone should have been impossible. Giant sponges, some approaching a meter in height, were clustered across the rocky seafloor in dense communities. Sponges grow slowly. Some species grow less than 2 centimeters per year. At that rate, a sponge a meter tall doesn't just represent decades of growth, it represents centuries. These were ancient organisms, old by any measure of marine life, and they had been living and growing in total sealed darkness, without access to surface nutrients, for longer than anyone could immediately calculate. Their very size was a clock, and the clock said these animals had been here for a very, very long time. Around the sponges, anemones with their tentacles spread in the current, solitary hydroids, relatives of jellyfish and coral that drift rather than form colonies, moving through the water. Brittle stars climbing across the sponge surfaces, using them as elevated perches to filter whatever nutrients the current carried. Deep-sea coral stalks rising from the sediment. An entire ecosystem built in layers, each organism occupying its ecological niche, with perfect, practiced precision. Then came the mobile predators, octopuses, resting on the seafloor at over 1,000 meters depth, watching the ROV's lights, with the calm intelligence their kind is known for. Icefish, Antarctica's cold-adapted species, carrying antifreeze proteins in their blood that prevent ice crystals from forming in their veins, even in near-freezing water. Sea spiders, not true spiders, but pycnogonids, moving across the rocks at sizes that shocked the team. Bodies the diameter of dinner plates, legs spanning wider. Crabs, amphipods, krill swarming around the ROV's lights the moment they switched on. And then, the creature that stopped the room, a giant phantom jellyfish, Stygiomedusa gigantea, one of the ocean's most rarely filmed deep-sea animals. The bell, the umbrella-shaped body, measured more than a meter across. Its 4 ribbon-like oral arms, the structures it uses to capture prey, stretched more than 10 meters in length. 33 feet of translucent tissue, drifting in slow, hypnotic pulses through water that had been locked beneath ice for centuries. Think about what it took to be that creature in that place. A giant jellyfish, filtering the current for nutrients in eternal darkness, completely cut off from the surface world, somehow finding enough to sustain a body the size of a small car. It should not have been there, but it was, and the question the team couldn't stop asking, as they watched it drift across the monitor, was a simple one. How? How did they survive? It is the central question of this entire discovery, and the answer challenges something so fundamental to how we understand life on Earth that it quietly rewrites the rulebook. In virtually every ecosystem on the planet, from the shallowest reef to the deepest trench, life ultimately traces its energy back to the sun. Photosynthesis at the surface produces plant material, which gets eaten again, which dies and sinks, raining down to the seafloor as what oceanographers call marine snow, a constant slow drift of organic particles, dead plankton, decomposing matter falling from the sunlit world above. Even at 2,000 meters depth, in total darkness, animals are eating particles that originated in sunlight. But this ecosystem had been covered by 150 meters of solid ice for centuries. There was no marine snow falling from above, no surface nutrient cycle, no photosynthetic base feeding the food chain from the top down. So what were all those sponges eating? What was the phantom jellyfish filtering? What was sustaining century-old organisms at the bottom of a sealed, lightless sea? The expedition's scientists believe the answer lies in ocean currents, specifically in lateral transport. The George VI ice shelf itself, as it slowly melts from beneath, releases glacial meltwater that carries something with it: suspended organic material, microscopic particles, dissolved nutrients, chemical compounds processed by bacteria living in and around the ice. These nutrients don't fall from above, they flow sideways, transported horizontally through the darkness by submarine rivers that move both toward and away from the Antarctic coast. Expedition scientist Dr. Laura Simile from the University of Cambridge explained it directly. The fact that they found long-living species suggests that lateral transport, mostly consisting of glacial meltwater from the George VI ice shelf, could be the source of the nutrients sustaining the thriving life found under the newly exposed ice shelf. What this means, in plain terms, is that this ecosystem evolved an entirely different feeding strategy from anything humans have studied in comparable environments. These creatures are not waiting for food to fall from above, they are positioned to intercept food flowing through the darkness horizontally. Their bodies, their feeding structures, their behavioral rhythms, all of it shaped over centuries or millennia by a current, not a sun. It is the ocean's version of a community that built its entire civilization around a river running through a cave, having never seen the sky. And once you understand that this is possible, once you accept that complex ecosystems can sustain themselves on laterally transported chemical energy rather than vertical photosynthetic input, everything changes. Not just about Antarctica, about where life can exist anywhere, the new species. In 8 days of exploration, ROV Sebastian documented 23 distinct megafaunal species. That number sounds modest until you understand what it represents, and what it almost certainly undercounts. Megafauna means large organisms visible to cameras. It doesn't count the microbial communities living in the sediment below the seafloor surface. It doesn't count the microscopic organisms colonizing the surfaces of every sponge and coral stalk. It doesn't capture the full scope of what was swimming through the water column between camera passes. It is, by definition, only the tip of the biological iceberg. Every species cataloged in those 8 days was the largest and most visible representative of an ecosystem that almost certainly contains hundreds of species the cameras never saw. Among what the cameras did catch, a suspected new species of isopod, a crustacean, a relative of the pill bug that Dr. Eschetti herself inspected after it was sampled from the seafloor. It may exist nowhere else on Earth, not in any other ocean, not in any other Antarctic location. Only here, in this sealed environment, shaped by hundreds of years of complete isolation into a form science has never described. The research team acknowledged it plainly. It will take scientists years to describe all of the new species found during this expedition. Laboratory analysis of the collected specimens will span multiple institutions and likely produce dozens of published papers. Some of those papers will name creatures that have never had names before. Antarctica's evolutionary isolation makes this especially significant. The Antarctic Circumpolar Current, a massive ocean current that encircles the entire continent, functions like a moat around a castle. Species that evolved in Antarctic waters have been cut off not just from surface ecosystems, but from the broader global ocean for millions of years. What evolves here alone, and what evolves beneath Antarctic ice shelves has another layer of isolation on top of that, cut off from even the Antarctic Ocean itself by 150 meters of frozen ceiling. These are animals that have been shaped by forces no other creatures on Earth have experienced. Their chemistry, their metabolism, their reproductive strategies, their immune systems, all adapted to a set of conditions that we are only now, for the first time, beginning to understand. The specimens being analyzed in laboratories right now may hold biological solutions to problems that scientists have been trying to solve for decades. Cold-adapted proteins, survival mechanisms for low-nutrient environments, novel chemical compounds produced by organisms that evolved in complete isolation. The practical applications of discoveries like these reach far beyond marine biology, but the full weight of what this ecosystem represents extends even further, beyond Earth entirely. What this means for alien life. For the last 2 decades, one of the most persistent debates in astrobiology has centered on a single question. Can life exist in an ocean sealed beneath kilometers of ice, with no sunlight, no photosynthetic base, and no connection to the surface world? The answer, delivered 1,300 meters beneath the Bellingshausen Sea on January 25, 2025, is yes. Consider Europa, the 4th largest moon of Jupiter, roughly the size of Earth's moon, completely covered by a shell of ice that is estimated to be somewhere between 10 and 30 kilometers thick. Beneath that ice lies a global saltwater ocean, kept liquid not by the sun. Europa is too far from the sun for that, but by tidal heating. Jupiter's immense gravitational pull flexes Europa's interior as it orbits, generating enough friction-based heat to keep an ocean liquid in conditions that would otherwise freeze it solid. That ocean has probably been there for billions of years. For years, scientists have looked at Europa and asked whether anything could be alive in that ocean. The theoretical case was always interesting. Tidal heating could drive hydrothermal vents on the seafloor. Hydrothermal vents could release chemical energy. Life on Earth has been found clustering around hydrothermal vents in the deep ocean. Ecosystems fed entirely by geological chemistry, independent of the sun. So, maybe, possibly, it couldn't be ruled out. But the skeptical counter-argument was always the same. We've never seen a complex ecosystem survive without any connection to photosynthetic energy. We don't know that it's possible. We're speculating. That counter-argument is now gone. We have seen it. A complex ecosystem, sponges, corals, octopuses, fish, jellyfish, sea spiders, dozens of species, surviving for centuries in a sealed ocean beneath hundreds of feet of ice, fed by nothing but chemical energy carried through the darkness by currents. It is not a theory anymore. It is a documented fact. And if it can happen in the Bellingshausen Sea, the physics and chemistry don't change on Europa. The same logic extends to Enceladus, one of Saturn's moons, which actively vents water vapor and organic compounds from its south pole, evidence of a liquid ocean beneath its ice. To Ganymede, Jupiter's largest moon, also believed to have a subsurface ocean. To Titan, whose hydrocarbon seas represent a completely different chemical template for life entirely. Every ice-covered ocean in the solar system just became a more credible candidate for harboring life. Not because the science changed, but because the experiment was finally run here on Earth, and the result came back positive. The search for life beyond Earth will never look quite the same after January 25, 2025. The clock is ticking. Here is the terrifying irony at the heart of this discovery. The same moment that allowed scientists to find this ecosystem is the moment that began to destroy it. For centuries, possibly for millennia, the creatures living beneath Iceberg 84 existed in a state of near-perfect stability. The temperature never varied significantly. The darkness was absolute and constant. The nutrient supply delivered by the lateral current flowing beneath the ice was predictable, consistent, the same day after unchanging day. The ecosystem had calibrated itself to those conditions with extraordinary precision. Every species there had evolved to thrive in that specific set of parameters. Then, on January 13, 2025, the ice was gone. Sunlight reached the seafloor for the first time in potentially hundreds of years. Not gradually, suddenly. The water temperature at the surface began to rise as open ocean replaced ice cover. The current patterns that had flowed predictably beneath the ice shelf began to shift as the physical structure that had shaped them drifted away. The chemical environment changed. The light environment changed. Everything that had been stable became variable, and variable conditions are precisely what highly specialized organisms are least equipped to handle. The team from RV Falcor 2 understood this immediately. That's why they moved so fast. Dr. Escate described it precisely. They seized the moment because they wanted to understand what was happening as the ecosystem transitions from being ice-covered to ice-free. Every sample they collected, every sediment core drilled from the seafloor, every specimen placed into a collection container, all of it was being gathered against a clock. The ecosystem they were documenting in late January 2025 may not exist in anything like the same form by the time a follow-up expedition could return. And this is not an isolated event. The Antarctic Peninsula ice sheet has been losing mass for decades. The peninsula is one of the fastest warming regions on Earth, and as global temperatures continue to rise, the frequency and scale of ice shelf calving events is increasing. More sections of ice shelf are fracturing. More icebergs the size of cities are breaking free. More sealed ecosystems are being exposed to open ocean conditions for the first time in thousands of years. Expedition co-chief scientist Dr. Sasha Montelli from University College London connected this directly to the broader picture. Ice loss from the Antarctic ice sheet is a major contributor to sea level rise worldwide. The work being done by this team, documenting these ecosystems, analyzing the sediment cores, understanding how the ice shelf has behaved over thousands of years, provides critical long-term context for the changes happening right now, and for the projections that will inform the policies that governments around the world are being asked to act on. What is being lost beneath Antarctica's retreating ice is not just ice, it is time. Millennia of accumulated biological history being erased by warming that is accelerating faster than science can keep up with. The sediment cores. Among the most important things ROV Sebastian brought back from the seafloor weren't the creatures. They were cylinders of mud. Long gravity cores, cylindrical samples drilled from the sediment, are geological time capsules. Each one captures hundreds or thousands of years of accumulated seafloor material in a single tube, with the oldest layers at the bottom and the most recent at the top. Every layer in that cylinder represents a different time period, a different ocean temperature, a different current pattern, a different biological community, a different climate regime. Reading a sediment core is like reading the rings of a tree, except the tree is the ocean itself, and each ring can span decades. The cores collected from beneath the newly exposed seafloor are currently being analyzed at research institutions across multiple countries. What they contain has the potential to answer questions that Antarctica's ice shelf has been sitting on for millennia. How long was this ecosystem actually isolated? The size of the organisms gives an estimate. Century scale at minimum, based on sponge growth rates. But the sediment layers will give a more precise answer, showing exactly when the ice shelf advanced far enough to seal this section of seafloor from surface conditions. It may turn out to be far longer than even the most cautious estimates suggested. How did the ecosystem respond to past climate fluctuations? Antarctica's ice sheets have grown and contracted through multiple warming and cooling cycles over the last several 100,000 years. Did the George VI ice shelf retreat during previous warm periods, exposing this seafloor to the surface world, only to re-advance and reseal it when temperatures dropped again? The sediment layers will show whether the biological community here has experienced disruption before, and if so, whether it recovered and how. What did the ocean currents look like through those cycles? The chemical composition of sediment layers records the temperature, salinity, and circulation patterns of the water above them at the time they were deposited. This is data that climate models need. Long-term, high-resolution records of how the Southern Ocean has behaved over thousands of years to accurately project how it will behave as warming continues. Dr. Montelli's team emphasized this explicitly. Understanding how ice shelves behaved in the past is essential for predicting how they will respond to future warming. The sediment cores from beneath the 84 are not just a scientific curiosity. They are calibration data for the models that will tell us how much Antarctic ice is going to melt and how fast, and what the consequences will be for coastlines around the world. The story of this ecosystem is inseparable from the story of what happens to every city built near sea level in the coming century. What comes next? The Falkor 2 expedition was not the first hint that life might exist beneath Antarctic ice. In 2021, researchers from the British Antarctic Survey drilled a narrow borehole through the Rona ice shelf, a different section of Antarctica's coast, and sent a camera down on a thin cable. The image that came back showed something unexpected: a rock on the seafloor, and clinging to that rock, faint but unmistakable, the outlines of what appeared to be stationary filter-feeding organisms, bottom-dwelling life where no life was predicted. But that was a borehole, a single narrow window. Whatever the drill happened to land on, that was all you could see. There was no way to know whether what was visible in that one small circle of light represented an isolated anomaly or the edge of something vast. There was no way to map it, to measure it, to understand the community structure, to collect specimens, to identify species. It was a hint, a tantalizing suggestion that something might be down there. The Falkor 2 expedition was the first comprehensive survey, the first time a remotely operated vehicle explored sweeping landscapes of seafloor beneath a recently calved ice shelf, filming in high definition, covering territory rather than a single point. It transformed a hint into documented fact, and in doing so, it opened a door that science will be walking through for generations. Because there are more ice shelves, Antarctica's coastline is lined with them. The Ross Ice Shelf alone covers an area roughly the size of France. The Filchner-Rona Ice Shelf is nearly as large. Between the two of them and the dozens of smaller shelves scattered around the continent, there are hundreds of thousands of square kilometers of sealed seafloor that may harbor ecosystems as complex, as ancient, and as biologically extraordinary as what was found beneath A84. Ecosystems that have never been seen, species that have never been named, biological strategies that science hasn't imagined yet. As climate change accelerates the calving of Antarctic ice, each breaking event creates a window, narrow, temporary, rapidly closing, into a world that has been sealed from human observation for longer than recorded history. The question of whether we will be ready to look through those windows when they open is not a scientific question. It's a question of funding, of political will, of how much a civilization values knowledge for its own sake. What the Schmidt Ocean Institute's team demonstrated in January 2025 is that the infrastructure exists. The ships, the robots, the scientific expertise, it's all there. What made this discovery possible wasn't technology that didn't exist before. It was a team that was already in the water, already doing the work, already positioned to pivot when the moment arrived. Being right there when this iceberg calved was extraordinary luck. But the lesson isn't to wait for luck. The lesson is to be in the water, to keep funding the expeditions that seem too remote, too expensive, too far from anything that immediately matters. Because the seafloor beneath the ice that just broke off the coast of Antarctica showed us something that rewrites our understanding of where life can exist on this planet, and by extension, throughout the universe. Giant sponges nearly a century old, coral forests in eternal darkness, a phantom jellyfish with 33 feet of tentacles drifting through a sealed ocean, 23 megafaunal species in 8 days, possibly dozens more waiting to be named in the laboratory specimens being analyzed right now. An ecosystem that survived for centuries without sunlight by feeding on currents in the dark, and in doing so, answered one of the oldest questions in astrobiology. Life does not require the sun. It requires energy. And energy, as it turns out, has a way of finding every crack in the darkness. Antarctica is still calving. The ice is still retreating. The windows are still opening. And somewhere beneath the ice that hasn't broken yet, something is still moving.